Welcome everyone. Today we're talking about human genetics, genome-wide association studies, and disease circuitry. And we're going to have a guest lecture by Jan Zhu on deep sea. So the goal of today is how do we interpret the genetic variation? So I wanted to start with a little bit of my own genome. This is actually my own genome. And I have 3 mutations in those 3 genes. And those mutations are shown here. And they're actually increasing my risk for age-related macular degeneration. How do we know this? By carrying out genome-wide association studies and then finding individual genetic variants that are associated with disease and that are differentially present in individuals that carry that disease and individuals that don't, enabling us to predict genetic locations that contribute to disease. So what we're going to talk about today is, number one, how do we even find these genetic variants in the first place? Number two, once we have found them, how do we figure out how they work? And how do we use them to then develop new therapeutics? So it's one of the most important areas of modern biology and modern medicine. And we're going to have a series of lectures on this. But today, we're introducing both the foundations of human genetics and the computational challenges in interpretation. So I'm going to talk about first human genetics, inheritance, Mendel, Fisher, single nucleotide polymorphisms, and other variants. Then about genetic gene hunting, disease gene hunting. How do we use linkage and genome-wide association studies to recognize locations that are associated with disease? Then I'm going to talk about linkage, disequilibrium, haplotypes, co-inheritance, and the challenges of fine mapping. Then I'm going to go through a case study of how do we actually understand the gene regulatory mechanism of noncoding genetic variants using the case study of this fat and obesity associated locus, which is the strongest association with obesity. Then I'm going to talk about how we associate at the large scale genetic variation with epigenomic signals that we have already studied before through ENCODE, roadmap, and more recently EpiMap. And then some machine learning tools for variant interpretation, including deep variant, a large set of predictors for the importance of individual regulatory regions, and then deep sea, for which we actually have a guest lecturer. So let's dive right in. Human genetics. This is a whole class on human genetics, compressed maybe in 5 minutes. So human genetics starts a long time ago. People have known for a while that if you selectively breed animals with more fur, at the next generation, you're going to end up with more fur. And we've ended up with corn instead of teosante, this like weird inedible plant in Central America. We've ended up with man's best friend instead of I'll have you for dinner. And people have long understood the inheritance patterns of eye and hair color and so on and so forth. And for a long time, people have hypothesized how inheritance actually happens, starting from ancient Greece and then continuing through the ages. People have understood that, you know, there's probably something with random mutation, with random mixing of traits, with natural variation, and then survival of the successful ones, which appears to be giving semblance to purpose. But unfortunately, Plato and the Stoics and, the more sort of religious view persisted of a betterment of the species and some kind of divine intervention. And this persisted until Lamarck and, up until Darwin, with the concept of transmutation and the concept that there's a complexity force that pushes organisms to become more complex. And then, of course, Darwin comes along and says, sorry, it's not that. It's instead random variation, standing natural variation that gets trimmed by selection. And then the surviving ones give the semblance of this driving force if the environment drives for complexity. And the field of genetics started roughly at the same time as Darwin, although the work of Mendel was not rediscovered for another 50 years, and many people couldn't reconcile the simple genetics that Mendel had shown in his peas with the complex genetics that were visible in humans. In humans, people had noticed this continuous variation in eye color, in hair color, in height, in skin complexion, in sort of so many different human traits appear to be varying in a continuum rather than discrete sets. By contrast, what Mendel was showing was the concept of particulate inheritance, that there was no blending, that there were discrete units of inheritance that would later be named genes, and that there were alleles that were dominant or recessive, and that a dominant allele would basically appear every generation, for example, brown eyes, whereas blue eyes, you would need to have a recessive allele come down from both lineages in order for it to be expressed only when both parents are in fact, the blue, give the blue allele to their children. And then this whole concept of independent assortment of discrete loci that would, sort of mix and match in, proportions predicted probabilistically. So anyway, this was the state of affairs, with Mendel having already worked out the rules of inheritance, but not being able to reconcile that with the biometrics of continuous variation. And that all changed in the turn of the century with Fisher and other statisticians that basically realized that continuous phenotypic variation could simply be explained by multiple Mendelian loci. So the notion is that, yes, height appears to be changing continuously, but even if you had only 5 genes for height, each of which would be pushing you by an inch up or down, it would appear to be a continuous distribution. That's a very important point. So I want to see who's following me on this one. The reconciliation between the discrete inheritance of Mendel and then the observed continuous variation in phenotypic traits could actually be explained by combining multiple, quote unquote, discrete loci, so-called Mendelian loci. So you guys are totally with me, 86%, 14%, 000. So this is the basis now for this past century's genetics that basically took us from understanding that multiple Mendelian traits could actually explain genetic continuous variation to understanding the molecular basis of inheritance, showing that chromosomes and DNA are probably what carries the genetic material, and ultimately solving the structure of DNA and understanding that that molecule, which is kind of boring in its structure, is in fact the basis of inheritance. And interestingly, while Mendel had struggled with deviations from his beautiful rule of independent assortment, this actually deviation became the workhorse of human genetics. How is that possible? Because what Morgan and Sturtevant showed was that traits that were near each other in the chromosome, and again, this was well before we understood the molecular basis of all that, but genes that are near each other in the chromosome tend to be deviating from independent assortment. If you're looking at, yellow-green seedlings versus, brown midriff, then these actually segregate independently and they follow Mendel's rules. But the closer these loci come to each other, the more they deviate. And the more they tend to be interacting, co-inheriting. And something is known as in equilibrium if it's far enough that they segregate independently. But if they are closer, they're known to be as in linkage disequilibrium. Okay, so who's with me so far with this concept that if two things tend to be co-inherited, that's because they're actually physically close in the chromosome. And if things segregate independently, assort independently, then it's because they're far from each other. Okay, great, 86, 14, 000. That's beautiful. Okay, so this basically became the basis for Mendelian trait mapping. So back in the 80s, we were able to already have a set of markers on the human chromosomes that are basically repeat length polymorphisms that allowed us to say what are the polymorphisms that each person carries, and then look for variation that co-segregated with these markers in order to be able to trace down the regions where different traits are encoded within the human genome. Okay, so the concept of linkage mapping is that if I want to figure out where is the gene that controls kernel color in my model system, or that controls cystic fibrosis in human, then I'm going to be able to see what it segregates with, and based on the fractions, I'll be able to actually map it down to a region of the genome. So who's with me here on the genetic mapping using linkage, using the segregation frequency of different traits with each other? That's awesome. It seems like all of you guys master genetics. So 90, 10, 0, 0, 0. That's awesome. All right. So that's the traditional approach. This is Mendelian mapping. And that's possible when the traits have a very strong effect, where you can say, okay, well, this person has cystic fibrosis, this person doesn't have cystic fibrosis. Let's figure out where they are on the chromosome and map them. The big revolution happened with complex traits that had up until now been impervious to this type of analysis and recalcitrant to this type of analysis. So in the 2000s, what we saw was the ability to actually map not just strong effect variation, but also weak effect variation in the human genome, and start understanding systematically those examples that I showed you, where my own genetic variants give me a slight predisposition to age-related macular degeneration. Here you have a twofold increased risk. This variant only gives me a 10% higher risk. So these variants would be unmappable with traditional linkage methods. So what we're going to look at today is how can we map them systematically using a genome-wide association study. And that's basically looking at every single SNP across 6 million SNPs in the genome, and how single nucleotide polymorphisms and how they vary with disease. And we're going to dive into GWAS very shortly. So let's start with the basics. What are the foundations of variation? Let's look at the different types of variation. We're going to be talking about SNPs, or single nucleotide polymorphisms, which are a single letter change. We're going to be talking about small indels, which are, TATGG is added to this position in addition to the C that's already there. We're going to talk about short tandem repeats, which are, GTC, 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 where we have about 1 every 10,000 base pairs. And then these large structural variants and copy number variants, which are in median length 5,000 nucleotides, which can basically insert or get deleted. And these happen roughly every million nucleotides. Okay, so who's with me here so far? So we have SNPs, indels, SDRs, structural variants, and copy number variants. Mostly we're going to be focusing on SNPs, but you should be aware of all of the other variation. So what do these do? Well, most of the time they don't do anything because they mostly fall in non-coding regions. But sometimes they fall either in a regulatory region and they have an effect, and we're going to talk about that at the end of the lecture, or they fall within the protein coding region and then they have a big effect. So if you look, for example, at this mutation here from A to T, and this box should be on top of it, this actually causes sickle cell anemia, where basically the blood cells sickle and they're not able to carry as much oxygen anymore. That's the bad side. The good side is that the blood cells sickle and they're not able to carry the malaria pathogen anymore. So sickle cell anemia is actually coinciding geographically with the locations in Africa where malaria is predominant, and that's very clearly due to selection for a mutation that makes it, harder to, function normally to sort of carry oxygen normally, but that also saves your life if the malaria pathogen happens to hit you. So these are the workhorse of genome-wide association studies, and also of something that we're going to focus on Tuesday, which is expression quantitative trait loci. So these are studies that look not at phenotypic variation relative to genetic variation, but also gene expression variation relative to SNP variation. Okay, so single nucleotide polymorphisms typically have two alleles. Basically, if you have the C allele or the G allele, these are the two different states that variable can take, and they're identified with their RSID. So you'll say RS189, 107, 123 is the SNP underlying sickle cell anemia. All of these variants have been clustered and built into a database, DBSNP, and there's more than 100 million known variants in DBSNP. And every now and then, they basically change the genetic code or they disrupt regulatory regions. Okay, who's with me so far on sort of the workhorse of genome-wide association studies, namely single nucleotide polymorphisms? Wow, we're hitting upwards of 90%. Wow, clearly going too slow here. So 95.5.0.0.0. Okay, so another type of variation is short tandem repeats. So you can see here, Huntington's disease is actually associated with this Huntington gene showing an increased length with, instead of having 9 repeats of CAG, 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 you can actually have more than 30 repeats. And then this causes this abnormal protein aggregation, and it damages neurons, it leads to brain cell death, and ultimately phenotypic effects like mood changes, coordination, speaking, dementia, etc. The next type of variation is insertions and deletions. So, for example, cystic fibrosis has an insertion of CTT, which basically, causes this loss of this phenylalanine, and then ultimately that leads to lung infection, cysts, and fibrosis, and extremely detrimental as well. All right, so we're going to be talking about reference and alternate alleles based on whether they match the reference human genome sequence, major or minor alleles based on whether they're more frequent in a particular population, ancestral versus derived alleles based on whether they match the most common recent ancestor between human and chimp, and then risk versus non-risk alleles based on their disease association. And then you should remember that some of them are environment specific. So for malaria, the risk allele is the opposite than for sickle cell disease. Okay, and then we're going to be talking about common, rare, and private variants. So common variants are more than 5% in the population. Low frequency variants are typically defined between 0.5% and 5%. Rare variants, less than 0.5%. And then private and de novo variants are typically the ones that either are private to a particular family or de novo since the conception of a particular individual. And then somatic variants are a subset of a person. Basically, these are variants that happen during your transition from a single zygote to a human being. During those cell divisions, you can actually have additional mutations where, for example, this part of my lung might have a G when the rest of my body has a C at that mutation because the progenitor cell that gave rise to that part of my lung has a G. Okay, who's with me on the number one, reference alternate, major, minor, ancestral derived, risk, non-risk, and then the frequency of common to rare to somatic variation. Okay, awesome. You guys are totally on the ball on this one. So 80-20-0-0-0. Okay, so we have a big challenge ahead of us. We have 2 copies of the genome in every one of our cells, 23 chromosomes, 20,000 genes, 3 billion letters of DNA, and millions of polymorphic sites. So how do we go about finding these disease genes? So the major concept that I want all of you guys to get is the difference between common variants and rare variants. So basically, what rare variants allow you to do is look at strong effect variation. So these are variants that, hold on one second, I'm looking for, I don't have a slide on that, unfortunately. So You know what? I'm going to find a slide. Common versus rare variants. It should be easy enough. There you go. Okay, so there's two types of variants. Oops, gosh. Oh, there you go. So everybody focus, this is super important. On one hand, if we for Mendelian analysis, what we're finding is that the genes that cause Mendelian diseases, the alleles that cause Mendelian diseases, are very rare and they have a very big effect. So they're very rare and they have a very big effect. By contrast, the alleles that are associated with genome-wide association studies are very common and they have very small effects. Is this a coincidence? Not at all. The reason for this inverse correlation is that if something has a very strong effect, if it causes a particular disease, then evolution will not let it rise to high frequency. Okay. By contrast, if something has a weak effect, then it's perfectly tolerable that it rises to high frequency. And the reason why we're looking along this axis is because we're simply underpowered to catch these parts. Namely, there's tons of rare variants that just happen all the time that have almost no effect. But those are just simply not captured by genome-wide association studies or by Mendelian studies. But up here, we basically have Mendelian studies that work well because there's a very strong effect. But the problem is that we just have to go and hunt families because these effects are so strong that these variants are simply not common. Okay, so who's with me here on the inverse relationship between common and rare variation and the two sides of the spectrum being tuned to linkage analysis versus genome-wide association studies? So one is the Mendelian analysis of one gene or two genes at most, or very small number of genes, and the other one is thousands of weak effect genetic variants. Awesome. So you guys are 90, 15, 0, 0, 0. It's awesome. Okay. So genome-wide association studies on one hand are looking at many, many variants of small effects, and these are polygenic disorders. Mendelian analysis of linkage associations are basically looking at single gene disorders, and these effects are very strong, but the variants are, the frequency of the variants is very rare in the population. These on one hand are mostly non-coding because that's how you get weak effects, by changing slightly the circuitry. And these are mostly coding because that's how you get big effects, by changing the protein directly. And more GWAS is gaining power and able to go into sort of, many, many combinations of effects. And we're able to go to weaker and weaker frequencies. And then linkage analysis also gaining power to basically be able to detect more and more, oligogenic disorders. But they're, very much tuned to these two sides of the spectrum. Okay. So with linkage analysis, we already talked about how we can have markers along the chromosome that basically tell you at what fraction of the chromosome are you. I'm at 13%, 48%, 54%, etc. And then you can map different traits to the markers that are closest to them and their distance to other markers, enabling you to pinpoint very precisely where they sit on the DNA. Okay, so that's linkage analysis. And the way you do that is pedigrees of families, where you basically say, okay, well, here's a marker that we're studying, here's another marker, and so on and so forth. And you can see how these are segregating with the disease. And in fact, this marker appears to be segregating with the cases. And therefore, my guess is that whatever gene causes that disorder, whose phenotype I'm following through the families, is in fact closely linked physically to that location of the genome. I went a little quick here, but I want to make sure that you guys are with me. So what linkage analysis does is that it has all those markers across the chromosomes. And then when it has a phenotype that it traces across the population, it asks which markers is it co-inherited with. And that's how you figure that out. Okay. Now I know that you guys are being honest because we're at 50, 40, 550. That's awesome. Okay. So Most of the workhorse of human genetics and most of the challenges computationally are here in how do we understand polygenic variation. The statistics for Mendelian analysis have been worked out for decades. And once you figure out the gene, there just aren't that many computational challenges left. But this is an area of immense investigation. So that's what we're going to be focusing on. So we're going to be looking at genome-wide association studies. What do these do? They basically gather thousands of individuals, some cases, some controls, partition roughly 50-50 traditionally. When I'm studying schizophrenia, I will go hunt for 60,000 cases and I'm going to find 60,000 controls. If I'm studying any kind of, like obesity or diabetes, I'm going to go and hunt 60,000 and find as many controls. So next Thursday, we're going to talk about the UK Biobank and large phenotyped cohorts where I don't have the luxury of having 50% schizophrenic cases. What I end up with is a fraction that is corresponding to the population prevalence. If 1 in 1,000, for example, carries the disease, then in my biobank, only 1 person in 1,000 is going to have that phenotype. Okay, so that's the case control design, where you should be aware that for genome-wide association studies, we're greatly overrepresenting the cases. So you've gathered a bunch of cases, a bunch of controls, and then you go and genotype them. And that's enormously important because we don't have to resequence every individual. Genotyping is extremely cheap. Sequencing the human genome cost us $1 per nucleotide. That's $3 billion. That's sequencing the first human genome. Genotyping a person at the same time was about $200. We're talking about $3 billion versus $200. For $3 billion, I wouldn't be doing 1,000 cases and 1,000 controls. But for $200, oh, sure, give me 10,000 cases, give me 100,000 cases. Okay? So genotyping technology is possible because we're able to catalog common genetic variation. We're able to sequence a bunch of individuals and then systematically catalog what are the common genetic variants in the human population, and then go and spot those variants with the G version here and the C version here, and then create probes that hybridize to the G version or the C version, depending on whether you carry one, the other, both, basically 2 copies of one, 2 copies of the other, or both, whether you're homozygous, let's say risk, homozygous non-risk, or heterozygous. Okay? Everybody with me on those 2 concepts of case control, overrepresenting the cases to gain power, but we don't have that luxury in biobanks, and genotyping instead of sequencing. And the reason for that is that it's immensely cheaper. Okay? So let's see who's with me on genotyping and case control studies. So what I have done is I've gathered 60,000 people with schizophrenia, 60,000 people without, and then I look at their common variants and how they differ. Okay, so we're at 50, 46, 0004. And then you do a lot of quality control. You look at sample quality, you look at relatedness, eliminate people who are related by descent rather than related. There's identity by descent and identity by state. These are the 2 concepts you should be aware of. And you don't want siblings in your study because that can dramatically boost the power of just random variants that these 2 people happen to have, even though what's causing them to have the same phenotype is the fact that, I don't know, they grew up in the same household or they share a ton of rare variants that their parents gave them. And then you want to correct for population stratification, and we're going to hear some about that by Alkis Price next Thursday. So you have a bunch of cases, controls, you correct for the fact that, Japanese individuals are here, and then, European individuals are here, and African individuals are here, and so on and so forth. And then you do a lot of QC for not just your samples, but also your SNPs. Basically make sure that the genotype quality is good, that it doesn't deviate from a normal distribution, and that the allele frequency is as you would expect. And then you basically, this is one of the types of QC where you do a quantile plot. You're basically plotting what would be the expected p-value if this was normally distributed. What does that mean? That basically means that if I sample 6 million SNPs, I expect, I expect, let's say, 1,000 SNPs to reach a nominal p-value of 1 of 10 to the minus 3. Okay, so basically I want to make sure that if I sample randomly, the expected p-value that I would expect based on the number of tests that I'm doing, in fact, matches the observed p-value distribution. And that I want to make sure it happens for the vast majority of the distribution, except for the places where the signal actually lies. This is where the actually truly associated single nucleotide polymorphisms are. Okay, so a QQ plot allows you to look for the presence of actual signal by looking at that p-value distribution. Okay, so now what is that p-value that we're computing? That p-value comes from a chi-square statistic. This is basically a table that tells you for every SNP in your genome, how many cases carried that SNP and how many carried that allele A of that SNP, and how many controls carried allele A of that SNP. So you're basically asking if there's an imbalance for that SNP, where the cases are not distributed in the same way as the controls. Okay, let's look at the numbers here. For allele G, you see that we have, 900 cases, 900 controls. Well, great, that's what I would expect here. But wow, wait a minute, I see way more allele A among my controls. So that suggests that perhaps allele G, anyway, that allele A is protective. Okay, so everybody with me? So I would expect uniform ratio, if you wish, between cases and controls for both allele A and allele G, but I don't see that. I see that cases have many fewer of allele A, whereas controls have many more of allele A. Okay, how do I measure the statistical significance of that? Well, I can basically look at a chi-square statistic that is basically telling me, given the 2 degrees of freedom that I have in this table, then I can look at a chi-square distribution with 2 degrees of freedom that basically, allows me to read off the p-value of that event. And the way that I calculate this is the observed minus expected square divided by expected. So that's how I get the chi-square distribution. And then I look up from that table, how many degrees of freedom do I have in my contingency table? And then that gives me the p-value for that. So if I have a chi-square of 25, this is way, way out there. And that's basically way, way close to 0. And that's a p-value of 10 to the minus 7. Who's with me here on how I calculate these p-values for genome-wide association study? So I take every SNP in the genome. I look at the frequency of A's and G's of each of the 2 alleles in case and controls, and I look for deviation in that frequency using a chi-square statistic, and then looking up the p-value based on the number of degrees of freedom of that matrix. Okay? All right, 62, 24, 14, 0, 0. Any questions, please type them into the chat. So now I can carry out that, and if anybody wants to unmute and ask a question, please do so. But basically, this is a very, very simple chi-square test that simply asks, is there deviation from what I would expect if there was no effect of that SNP on the phenotype? And if there is an effect, that basically means that the probability with which I would expect to see that distribution by chance is very small. And that's what the p-value is telling us. The p-value is basically saying that we can reject the hypothesis with which, the hypothesis according to which the allele has no effect on the phenotype. We can reject it with certainty of 10 to the minus 7, that I would have expected that big of a chi-square test value only, once in a million times. Okay. So these are the p-values that I'm now going to compute across the genome, and I'm now going to plot in a Manhattan plot. So what a Manhattan plot is telling us, named after the skyscrapers in the town of Manhattan, what the Manhattan plot is telling us is what is the negative log 10 p-value of that chi-square statistic. And that chi-square statistic is computed at every SNP in the genome. And then I'm asking how many of the SNPs have a very, very small p-value, 10 to the minus 10, 10 to the minus 13, and so on and so forth. Okay, who's with me here on the Manhattan plot? So what a Manhattan plot is telling me is the minus log 10 p-value of the probability with which that SNP is randomly associated with the disease. And if that p-value is enormously, that minus log 10 p-value is enormously high, if that p-value is enormously small, it basically means that that SNP is non-randomly associated with the disease. All right, 74, 21, 5000. All right, so after I have my Manhattan plot, I can take the same p-values and plot them on a QQ plot. And then I can do all kinds of additional analysis by combining multiple studies. I can say, well, in Denmark, they did a study with schizophrenia. And in the US, they did a study with schizophrenia. And in Asia, they did a study with schizophrenia. So I can now combine all of these to basically have a combination of cohorts that then allows me to evaluate with more confidence the association of every SNP. And then the next step is how do we do functional analysis to understand what the role of these SNPs is. For example, I can go and perturb that SNP between A and G and then see if it changes luciferase activity for enhancer, basically measuring whether that SNP in fact perturbed the enhancer. And we're going to talk more about that throughout the term. And of course, I can do other analysis. I can look for gene-based analysis. I can look for pathway analysis, polygenic risk estimation, SNP-SNP interactions. And we're going to talk about a lot of that on Tuesday next week. All right. The challenge, of course, is that we are testing millions of SNPs. We are testing every single SNP in the genome. And therefore, if I see a p-value of 10 to the minus 7, and I've tested of 7 times 10 to the minus 7, which is basically almost 10 to the minus 6, and I've tested 10 to the 6 elements, then maybe I shouldn't be that excited. And that's where genome-wide significance comes in. And that's where this red line comes in. This red line basically tells me, given the enormous number of tests that I'm carrying out, I'm not going to get excited unless something has a 10 to the minus 8 p-value. Okay, or 5 times 10 to the minus 8, so slightly less than 10 to the minus 8. So slightly less significant. So that's what we define as genome-wide significance. And this was basically based on a back of the envelope calculation 20 years ago that basically said, well, we better be a little more stringent because a lot of our people are reporting nominal significance and, the field is starting to not trust us. So we should, based on there must be roughly 1 million independent SNPs in the genome. Let's do a back of the envelope calculation to basically say 0.05 divided by a million tests. Well, that's 5 times 10 to the minus 8. And that's how this number came about. And it turns out that number was actually quite accurate after a lot of revisiting later on. Okay. So we've talked about Manhattan plots. We've talked about QQ plots. And the next thing is fine mapping. Okay. This is challenging. The reason for that is that the human population is still a very small population. We are, enormously populous since we came out of Africa and took over the world, but the genetic variation that we have is still that of a relatively small population. And that means that there hasn't been enough time for all of our SNPs to segregate independently. There are only a small number of recombination hotspots in the human genome that have been, broken up over the generations. And that basically means that when this variant is associated with the disease, then a bunch of other variants nearby that are co-inherited with that variant are also associated with disease. And then after I go through this recombination hotspot here and this other recombination hotspot there, then there are no other variants that are co-inherited with that one. And therefore, the signal of this causal variant doesn't leak out outside these linkage disequilibrium block boundaries. Okay, so let me recap, and then we're going to dive into that again in a few moments. But there are only a small number of recombinations that happen in the human chromosome across any one generation and across all of our generations, and since the out of Africa event. And that basically means that if I have one variant in that region, if I have the A allele in this region, I can probably predict that I also have the, let's call it the A allele here, the A allele there, the A allele there, and the A allele there. Okay. So if I have inherited the, I don't know, Irish version here, then I have inherited the Irish version everywhere. But if I have inherited instead the Italian version, then I have the Italian version everywhere. Okay. So who's with me here on the fact that variants are not inherited in isolation, but they're inherited in blocks? And therefore, if one variant in that block has the Irish allele, then every variant in that block has the Irish allele. Okay. Okay. Very cool. So, and therefore, what we need to do is after we found the association in that region, we need to now go and say, okay, well, but which SNP, which single nucleotide polymorphism in that region is in fact responsible for that association? We're at 55, 41, 5, 0, 0. So 95% of the people are 60% or above, which is great. Okay. So let's take a quick break here and see who feels that they've learned something. Okay, good. So we're at 48, 48, 0, 0, 0. This is great. And then 4. Okay. So, quick poll on pace. Am I going too fast, too slow, or just right? I'm sure it's too fast. Oh, just right. Wow. Okay. So the same person who voted that they haven't learned anything are also saying, you're doing way too slow. Anyway, so just right, 68%, slightly too fast, 27%, and then way too slow, 1%. Okay, so let's dive into some results. So this is one of the first genome-wide association studies for Crohn's disease, carried out by Mark Daly and his colleagues here at the Broad Institute. So what did it find? It found one region here. You can see all of these dots that rise up way above genome-wide significance. Why are there so many dots? The reason is that when you have a linkage disequilibrium region and they're all associated to various degrees, you squish it, when you're showing the whole genome, you basically see that all of these other SNPs are basically lining up on top of each other. Okay, so you have one such region here, another such region here. Oops. Hold on one second. Mon amour, National Grid is calling. Can you please talk to them? I'm giving my lecture. Hold on one second, Barbara. My wife is coming up the stairs. When the gas company calls, you answer. You basically have two different examples, one of which was also discovered by linkage. The other one was not discovered by linkage. So that's kind of interesting, right? I was telling you that GWAS is on one side and then linkage is on the other side, but linkage analysis also discovered this region, and then genome-wide association studies only discovered that region. Why is that? If you look at the one that was also found by linkage, not to card 15, it's at 5% frequency and it has an effect size of 3.0. What does that mean? That means that if you carry the risk allele, then you are 3 times more likely to have the disease, to have Crohn's disease. By contrast, IL-23R, which is here, is actually a protective allele. This allele decreases your risk. So the rarer allele, the less common allele, decreases your risk. What does that mean? That means that when I do my case control cohort and I'm enriching greatly for cases, I can basically only find risk increasing variants because I'm dramatically enriching for cases. I will rarely find protective variants. Okay? These are some subtleties, but the linkage pedigrees that you would need to achieve significance for this one, which is protective, would be 30,000 people, 30,000 families. And that's just simply not feasible. And if you look at ATG16L1, which is, and this one here that barely passed genome-wide significance, ATG16L1, then it is a risk allele, but it only increases your risk by 40%, as opposed to 300% for this one. And for this one, to discover the small effect of a 40% increase, you would need 40,000 pedigrees, okay? So that's why linkage analysis and GWAS actually act at different regimes of common versus rare. And that's sort of the famous slide that I wanted to show you of, on one hand, you have the common variants that GWAS captures. On the other hand, you have the rare, strong effect alleles that linkage captures. Okay? All right. So now we talked a little bit about haplotypes. Let me give you just a little bit more background. So again, the way that we think about haplotypes is as you walk along the genome, you basically can ask what is the correlation structure between all of these different SNPs laid out side by side. And that correlation structure tells you that all of these SNPs are co-inherited. And between them, there's a linkage, there's a recombination hotspot. And all of these SNPs are co-inherited, and then another recombination hotspot. And all of those are co-inherited, another recombination hotspot. All of those are co-inherited, another one here, and so on and so forth. But it's not just super discrete. You know, there's a weak one here, but then there's strong correlation there too, and so on and so forth. So basically, it's not a yes, no, it's just a frequency of recombination, okay? Who's with me on the linkage disequilibrium hotspots here and how they work on the genome? Oh, sorry, sorry, wrong poll. Up, are you falling so far? There you go. Okay, lovely. Cool. So 90% of people are 60 or above, which is great. Okay, so that's basically recombination, and then we can quantify it all kinds of interesting ways. And then it varies across populations. You know, the chromosomes that went to Africa are slightly different, sorry, that, you know, sort of continued populating Africa versus the chromosomes that went to Europe or the chromosomes that went to Asia are very different from each other. And then you can have different recombination hotspots in each of them. And then there's a mechanism of recombination that leads to these hotspots. And this mechanism of recombination is guided by PRDM9 that basically goes and finds a motif and then creates a double-stranded break during meiosis for the chromosomes to line up and for every gamete to have a correct number of chromosomes, okay? And these double-stranded breaks and these strand invasion and sort of recombination are key for lining up chromosomes, and that's also how you get recombination events, okay? All right, so we've now discovered an association. How do we figure out how it works? So I'm going to tell you about a study that Melina Clausentzer and many other colleagues, including, you know, several folks from my lab, were involved in and was published a few years ago in the New England Journal of Medicine. And this was perhaps the most comprehensive mechanistic dissection of a non-coding disease locus, okay? This is starting from a SNP, from a single nucleotide polymorphism that is associated with disease, and then asking how does it work, okay? And then the 6 steps that one needs to take is what we took here. So let's dive into the SNP first. So this is the genome-wide association study results for obesity or body mass index. body mass index is a measure of obesity. And back in 2007, this locus here, which was promptly renamed Fat and Obesity Associated, FTO, was in fact found to be the strongest genetic association with obesity. So people basically said, okay, there's something important here. And when you open up the hood, as I showed you earlier, when you look at all of these SNPs and you line them up side by side, you see that there's 2 recombination hotspots here, but there are 89 common variants in this 50,000 nucleotide region that are all co-associated with disease. So that's the challenge ahead of us. How do we make sense of how does this thing work? We know that there's something going on in this region, but we don't know how it works. So what are the steps? The first step is we need to figure out where does it act? What is the relevant tissue and cell type? The second challenge is how do we find the downstream target gene? Because these are non-coding variants. Maybe they affect the expression of FTO, but maybe they affect the expression of some other faraway gene that is hidden. And in fact, they do. How do we find which variant among the many that are in this region is the causal one? Namely, if I go and genome edit this one or that one or that one, will I restore the phenotype, or do I need to sort of find the correct one to do so? How do we find what is the upstream regulator that controls that region whose binding is disrupted by loss of that variant? And number 5 and 6, how do we establish the cellular and the organismal phenotypic consequences of that of those target genes? So who's with me on at least the challenge ahead, the 6 questions of where does it act, through what gene, via what variant, what regulators affected, and then what does it ultimately do at the cellular and at the organismal level? So great, 100% of the people are above 60%, great, 53, 47, 000. Okay, so how do we do that? The first challenge is the tissue. And that's where we basically looked at the epigenomic annotations of 107 different tissues across the Roadmap Epigenomics project. And we found this super enhancer, this 12,000 nucleotide enhancer control element. Enhancers are normally very small, 400 nucleotides, maybe 1,000 if you're, really lucky. But this is 12,000 nucleotides. It's a master regulatory control region. And it sits exactly in this region of association, and it acts in the progenitor cells of white fat cells and beige fat cells. It's associated with obesity, and it turns out that it decides whether your fat cells differentiate into fat storing cells or fat burning cells. Guess which one's associated with obesity? The fat storing one leads to obesity, and the fat burning one leads to sort of lean individuals because they burn all their excess calories. Okay, so that's where this enhancer acts, but we don't yet know that the SNP has anything to do with that process. The second step, as I mentioned, is to find the target genes. And here I'm showing you the recombination hotspots of the human genome and all of the genes nearby. And you can see that the genetic association is very tightly localized to only one gene, FTO. And the pharmaceutical community was super excited. It's like, great, there's only one gene here. We know what gene it is. But the problem is that gene regulation doesn't care about recombination hotspots because of the long-range linking and long-range looping that we talked about a few weeks ago. So in fact, that region containing the FTO genetic variant association is linked to a bunch of other genes, both to the right and to the left, suggesting that any of them could be the targets because they're forming these loops. So the next challenge was, how do we figure out what genes among all these candidates change in expression between the risk and the non-risk allele? So we basically went and measured the expression of homozygous risk and homozygous non-risk individuals for each of these genes. And what we found is that the FTO gene itself had no change in expression. And instead, this gene here, which is many, many recombination blocks over, and that gene here that actually sits at 1.2 million nucleotides away, all the way out here, several genes away, are in fact the only two that change in expression, suggesting that IRX3 and IRX5 are in fact the target genes. Who's with me so far on finding the target genes for these non-coding loci? Awesome. Wow, you guys are really with me. Notice the risk allele actually increased expression. 63, 37, 000. The risk allele increased expression. So this is a gain of function. And that's something to realize, that most of the time we're saying, oh, that gene is deactivated with the disease. But no, these two genes are hyperactivated because the repressor can no longer bind. The next challenge is understanding what is the causal SNP. And there we use regulatory motif analysis and evolutionary conservation, and we're able to pinpoint a single nucleotide alteration that disrupts this evolutionarily conserved AT-rich motif with a C at that position for the risk individuals for obesity. And we went and tested that if we change that SNP at the cis level, and if we change the upstream regulator at the trans level, then we can actually show an epistasis between the two. Let me explain. This AT-rich motif is recognized by this AT-rich interacting domain protein 5B, RE5B. We basically said, can I knock down the upstream regulator, and can I change the SNP between the risk and the non-risk individuals? And it's only when you have the correct motif and the regulator being active that you see repression. And if you disrupt either the upstream regulator or the SNP, then you see de-repression. So there's an upstream regulator that normally binds that motif, and when the SNP happens, the repressor can no longer bind, and enhancer is overactivated, and IRX3 and IRX5 are overactivated. So both at the enhancer level and at the gene expression level, you get repression only with the intact motif and the intact regulator. This is more complicated, but let's see who's following me here on the cis-trans analysis. We have the motif, we have the upstream regulator. If we disrupt both, then we have de-repression. If both are intact, then we have correct repression. Okay, as expected, it's a little lower, 30, 57, 490. Okay, and then the next step is great. We have the circuitry. We have the tissue, pre-adipocytes, to decide whether you go to fat burning or fat storing. We have the target genes, IRX3 and IRX5. We have the causal nucleotide based on the genome editing, and we have the upstream regulator, RE5B. How does it actually function? Then we need to actually look at the cellular consequences. And what we found is that IRX3 and IRX5 are negatively correlated with mitochondrial function and positively correlated with lipid metabolism. And indeed, the risk individuals have fewer mitochondria and they have bigger fat cells, suggesting a shift from energy dissipation to energy storage. And we basically were able to build our model to say that we were able to go from a region of association where we knew nothing to having the SNP, having the motif, having the upstream regulator, having the downstream target genes, and having the biological process, which basically meant that we could go in and intervene and change that circuitry and show that by knocking down the downstream target genes, we could shift back and forth between lean and obese phenotypes. By creating adipose-specific knockdown of IRX3, we were able to change mice and shift them from regular weight to super lean by changing that gene to not be expressed. And then these mice were unable to gain weight in a high-fat diet. Normal mice gained weight. These mice were unable to gain weight. And with genome editing, with CRISPR-Cas9 genome editing, we're able to show that changing a T into a C and changing that C back into a T was able to change the expression of the downstream target genes to be up and then down again. So from the AT-rich motif, I disrupt the motif, the genes are de-repressed, the repressor can no longer bind. I restore that motif, the genes are repressed again. And then we can go from the risk allele to the protective allele by changing that C into a T, restoring the AT-rich motif, and then boom, the process of thermogenesis measured by oxygen consumption rate is in fact completely restored at the cellular level. So that allows us to now understand the mechanism underlying this SNP, okay? So I like to say that we wrote a paper about 1 bit of information in the 3.2 billion nucleotides of the human genome. 1 bit of information, this RS1421085 mutation from a C to a T, shifting between fat burning cells and fat storing cells, okay? So the challenge ahead of us is that we want to do this another 120,000 times. And we want to develop computational methods for doing that systematically. So that's what we're going to hear about today from our guest lecturer, who I hope is here.
Yes.
Oh, perfect. Jan is here. Hey, Jan, thank you so much. Can you share your screen and take it away? And then on Tuesday, we're going to have a lot of additional information on how do we do this systematically at the systems level. So Jan is going to be telling us about how we can use the deep learning techniques that we heard about already to now start predicting the impact of SNPs, 120,000 SNPs at a time, rather than 1 SNP at a time. Jan, take it away.
Okay, thank you. So I'm Jian Zhou. I'm the system.
Oh, I cannot hear you. Your microphone must be off because you're not muted.
Can you hear me now?
Nope, still nothing. Oh, you can hear him, but quietly. Okay, let me boost up my volume. Oh, my volume is off. Okay, awesome. Jan, they can hear you. I can't.
Okay, so it's fine, right?
Yeah, perfect.
Okay. Okay, great. So I'm Jen. I'm an assistant professor in UT Southwestern. I'm glad to talk to you today about some of our works on deep learning models of genomic sequence, and especially in consistent with today's theme, I'll be focusing on the applications of those models to variant effect predictions. So I'll start with a little bit of motivation of why we're interested in studying the problem. And first of all, every one of us has a different genome, right? And the genome can give us different traits and may predispose us to different diseases. And there are numerous differences in between any two genomes. And there could be even differences that are mutations that are only unique to you, to one individual. Therefore, it is very difficult, if not impossible, to use experimental approaches to study all of those individual genome variations. So therefore, we would like to have an approach to computationally predict the impact of those genome differences. And on the other hand, of course, the same genome gives rise to all of those different cell types. And the same genome has to be differentially utilized in different cell types to regulate those expression and other processes in different cell types. And of course, there are tons of studies about this, but still, we are not at the level that we can say, like, given a sequence or given a particular mutation, what would be its impact in all those cell types. So we would like to gain a better and better understanding to how does genomic sequence and mutations and variants of genomic sequence affect all of those cell types with specific regulations and their impact to human health. So we take a machine learning approach to utilize our genomic sequence and the large amount of functional genomic data that are available to build models that can predict the effect of genomic sequence and sequence variations. So I'll start from the very beginning. So the very first problem that we look at is what we consider as the first step of genomic sequence-based regulation, which is how does genomic sequence decides the binding of individual transcription factors and subsequently the organization of chromatin, including the histone modifications and chromatin accessibility. So we want to be able to do, like, we want to train the model to predict from genomic sequences across the genome and predict the binding of different transcription factors and histone modifications and chromatin accessibilities at the center of this sequence across different cell types. And at the time, there wasn't any deep learning models for genomic sequence developed yet. And the reason that we chose to use a deep learning model, specifically deep convolutional network model, is mainly because it satisfies three of our requirements that we want to build into our regulatory sequence model. So first of all, we would like the model to be able to use large sequences and long sequence context, because it has become clear that if you just look at a single motif, it's not sufficient to accurately predict the TF binding. So we really like to be able to utilize the sequence context. And secondly, because we would like to utilize a large sequence context, to use that efficiently, then we have to be able to model the nonlinear interactions across different regions of the sequence. And thirdly, because we are predicting all of those different transcription factors and chromatin properties, what we would like to be able to share sequence features that are learned across all those tests, because the sequence patterns that are predictive for the binding of one transcription factor can also be useful for predicting the binding of another relevant interacting transcription factor. And also all of those proteins and histone marks are interacting with each other. So things that are informative to one could be also informative to the other. So you want the model to be able to share information from each other. So a deep convolutional network model can give us all three of those features. And indeed, it turns out to work very well in making those predictions. So I'll just give a quick flyover about the actual design of the model, because I realized that you have already learned about several of the convolutional network model-based convolutional network-based model of genomic sequences. So this was new at the time, but now there have been a lot of studies have been using convolutional network like structure to study the genomic sequence. But the basic principles are still the same. So in convolutional network, we use convolution layers to learn different levels of sequence features. At the lower levels, you can consider it as learning the equivalent of motifs. And at the higher levels, it learns the higher order sequence patterns, such as you can consider maybe like a motif or motifs. So those longer sequence patterns. And to be able to learn those hierarchical multiple levels of features, we have to build in a spatial scaling mechanism in the network. So you can achieve that by using the pooling mechanism, or you can achieve that alternatively by introducing dilations in your convolution. So either way, you need to introduce some kind of spatial scaling to be able to learn those larger scale features. And at the top, our network always pulls the information across the entire sequence. We want to combine the information from the whole sequence. And there are many different ways that you can do that. But just one thing to remember is that if you want to make position-specific prediction, for example, in our case, you want to predict the effect right at the center of this sequence, then you want to preserve the spatial information. So you don't want to use some layers like global max pooling, which will erase your spatial resolution. So if you want to make predictions that are position specific, then you want to use some mechanisms that can preserve the spatial information. So now with this model trained, so we have actually a pretty good model of predicting those transcription factors and histone modifications. And what can we do, right? So even though the model is trained on genomic sequences across the genome without seeing any genome variants, we can actually ask the model to make prediction for any sequence, and which means that we can use it to predict the effect of any genomic variant. For example, in this case, we want to predict the effect of changing this nucleotide from A to C, then we can give the model two sequences that only differs by this one variant, right? And then we can look at the two sets of predictions from the model for each of those variants. And if we predict that for the allele A, we have a high binding of this transfer factor, while we have a much decreased binding for allele C, then we are predicting that this variant actually decrease the binding of this transcription factor, right? So now the question is, how do we know that if our prediction is accurate, right? So at the time, there was not many, actually even now, there are not many high quality examples of variants that change the effects at chromatin level. So to address this, we actually look into those very deeply sequenced genomic data sets. Specifically in this example, we look into the very deeply sequenced DNA data set in about 30 cell types. But then in those deeply sequenced DNA-seq data sets, we look for positions where there are heterozygous variants in the sample. And for those heterozygous variants, then we look for whether one of the allele is significantly more represented in this data set than the other allele. So for example, if the reference allele is more represented in this data set, this may indicate that this allele could be more DNA sensitive than the alternative allele, and vice versa. And with this, we can get a large number of putative variants that has an effect on the DNA sensitivity. And we can now also ask the model to make this prediction. So we can just give the model the sequences of that carries this variant and predict the DNA sensitivity for the reference allele and the alternative allele. And with the model prediction, we can now compare with our experimental results, right? So we visualize it using this plot, where in the x-axis, we plot the prediction for the alternative allele, which is in probability scale, so it's from 0 to 1. And on the y-axis, I plotted the probability for the reference allele. And each of those dots is a variant. And now we're going to compare this with the, I'm going to color this with the experimental result. So if our prediction is correct, then those dots that are off diagonal are reference allele biased, which means it's more sensitive, DNA sensitive for the reference allele and should be labeled blue. And those that are below diagonal should be more alternative allele biased and should be labeled red. And this is indeed the case. And especially if you look into this plot, we have higher and higher accuracy if you look for those variants that has stronger difference between the prediction for the reference allele and the alternative allele. Or when we're filtering for predictions that are most confident, we can see higher accuracy of making such predictions. So we can quantify this performance using a curve like this, where the x-axis is essentially the distance to the diagonal. And on the y-axis, we see the accuracy of predicting which allele is more DNA sensitive. And each of those lines here is one cell type, and the bold line here is the average across all 35 cell types. And this evaluation actually has one feature that I didn't tell you, that it is actually robust to having false positives in our standards. So when we're detecting those reference allele and alternative allele biased variants, we're not guaranteed that every one of those variants that we detected are actually having a causal impact. So there could be false positives in these data sets. But if our model is good, then it is able to filter for the true positives when we set a threshold to filter for variants that are strongly predicted to have a strong effect, right? So if the model is good, then it is able to pick out the true positives and predict its directionality. And if the model is not able to do that, then it's not going to have a high accuracy. So this allows us to evaluate this with even those that are imperfect gold standard. And we also did this for histone mark QTLs. And which again are variants that are associated with histone marks, but not necessarily causal. And we can see that we can also predict the allele that's linked with higher histone marks. And finally, for the transcription factors, we didn't initially do this evaluation because the ChIP-seq data sets that measures transcription factor binding are generally having lower coverage. But actually later, an independent group also did this evaluation to show that DeepC is actually also pretty good at predicting the allele imbalance in transcription factor data sets, even though the measurement is noisier. So now with this capability that we have evaluated, that we have the capability of predicting the effect of those variants, right? And now we can, of course, apply it to predicting any variant, including those that we have never seen. So for example, for a single sequence, we can introduce all of the possible single nucleotide mutations to every position of this sequence and predict its effect. And I call this approach in silico mutagenesis. And with in silico mutagenesis, we can just look at the sequence like this. So we know that those mutations at those positions will decrease the binding of this transcription factor, while some other mutations will increase the binding of this transcription factor. And we can apply this to several different known pathogenic mutations. And what we see is that not just the known mutation has the impact that's reported in affecting this transcription factor binding. We also predicted many other mutations that are surrounding this site that has similar or even stronger effect. So that means that if maybe in future that we have a patient with the same disease, but having a different mutations that cause the same effect, even though it is not the original mutation, we may be able to predict that this mutation may cause the same disease through the same mechanism, right? So Manolis, how much time do I have?
Well, normally the class ends at 25, but we typically, I would say 100% of the time, go until 30. I think going past 30 is probably not a good idea.
Okay, so I'll aim to stop at 30.
Perfect.
Okay, so now I'll tell you another example of molecular level effect prediction, which of course, after we have been able to predict the effect at the chromatin level, we would like to be able to predict whether we can predict the effect of a variant at gene expression level. And there are many challenges on this. And I will not say that this problem is solved. It's far from being solved, but this is our first attempt to address this problem. And actually two of the challenges of predicting gene expression are, first, that it depends on much larger regulatory sequences. So you need to consider much larger genome region. And secondly, there are actually fewer training samples available. So we consider the prediction of gene expression, then we're limited by the number of genes in our genome. So we don't have an unlimited number of training samples. So we have to be smart about how to construct our features, not to overfit to this data set. So to do this, to address the first problem, we actually just look at a broad region of 40 kilobase pair, and we apply a pre-trained model of chromatin level prediction model to predict at every different positions of this window using a sliding window. So we actually predicted something like a spatial patterns of every chromatin profiles. And now with this spatial pattern of chromatin profiles, we want to train a model to predict gene expression. And in this model, we actually just use a regularized linear model that learns the contribution of each chromatin profile to the gene expression at each different positions. So we learn these smooth patterns of contributions from each gene expression profile prediction to the gene expression. And how do we do that? With a linear model. So each of these curves that you're seeing is actually a linear combinations of basis functions, exponential basis functions. And to do it, it's actually pretty simple, that we can transform our spatial patterns here. And now I represent it as a matrix, and each columns here is a different spatial positions. And I transform it by multiple exponential basis functions So 5 for downstream, 5 for upstream. And we can compute the weighted sums of those spatial features for each one of those exponential basis functions, resulting in multiple summarized spatial features. And now, if we train a linear model on top of those linear spatial features, we are actually learning linear combinations of those exponential basis functions, which allows us to learn the smooth patterns about the contribution of those chromatin profiles to gene expression. And of course, those chromatin profiles are also predicted from the sequence. So now we can do the same trick that we can predict the effect of any variant on the gene expression. And we can also evaluate that using a similar approach. So now we're using the data about eQTLs, where we know that which variant is associated, which allele of the variant is associated with higher expression. And we also see that we can pretty accurately predict all of those eQTL directionalities using our approach across multiple data sets. So I think given time, I'll actually skip over the rest of this. And
I would say I would love to hear about the autism variants, especially because I think the rare end of the spectrum is something that is particularly interesting.
Okay, so I just give you one more story about autism.
Great.
Right? So we'd like to apply our ability to predict the effect of genomic variants to actual human disease data, right? So one of the applications we consider is whether those de novo non-coding mutations contribute to diseases such as autism. So de novo mutation is a family of mutations that are generally ultra rare in human population. It's a mutation that are only in you, but not in any one of your parents. And we generally know very little about those de novo mutations, especially for de novo non-coding mutations. So it has been difficult to attribute whether those non-coding mutations are contributing to disease or not. So in the case of autism, that we have previously known that the de novo coding mutations is the major contributor to autism. And much of our knowledge now about autism are learned from the coding mutations. But for non-coding mutations, it's been difficult to detect a contribution. And so this is the data that we have. So we have those autism mutations, de novo mutations from the simplex families, which means that we have an autism individual and another individual without autism, which is our controls. So we can have mutations from both the proband, which has autism, and an unaffected sibling. And then we can compare these two sets of mutations to see whether there is a significant difference, right? And if we just compare those two sets of mutations by number, it's actually very hard to see a difference, even if you subset to different genomic regions. So we think that maybe we can look at this differently now that we have the ability to predict the effect of those mutations. So me and my collaborator, Chris, trained those two sets of deep learning models. So one is similar to DeepSeek that I talked about, which predicts the chromatin profiles, and another is trained to predict the iron binding protein binding profiles, which predicts the effect at the RNA level. So now with these two models, we can actually summarize the effect of mutations to a single score at both DNA level and RNA level. And now we can compare the proband mutation and sibling mutations to see whether proband mutations is having a stronger impact than sibling mutations, right? And this is indeed what we saw. So we saw this for both proband, for both DNA level and the sibling and the RNA level, that we see stronger effect in proband compared to sibling. And we see even stronger effect if we focus on gene sets that are previously known to be relevant to autism through coding mutations. And these are just selected gene sets. So here I'm showing you all of the gene sets that we have considered in this study. So across all of the gene sets and with multiple hypothesis correction across all of those tests, that those gene sets are the ones that are significant after the correction. And we see that the strongest ones are, for example, the brain expressed genes that are affecting the the RNA binding protein binding. And also for all of those categories that are significant, we see stronger effect in proband rather than sibling, which is also confirming that we are detecting a contribution from those mutations to autism. So lastly, we also want to be able to do some unbiased analysis. So here we're using the gene sets that are previously known, and we wish to be able to see the contribution from non-coding mutations in a relatively unbiased way. And this is challenging to do at the gene level because we lack a statistical power. So it is not possible to say that at a single gene level that we see stronger effects in proband compared to sibling. So we did a network neighborhood based analysis. So now we're looking at so given a gene network we have already had from prior studies that tells you about the functional relationship about the genes, we can now look at for a single gene and in its network neighborhood, whether we have stronger effect proband mutations compared to sibling mutations. So we can do a weighted test that's weighted by the network connectivity of all the neighborhood genes to the gene of interest and compare the proband and sibling mutations. And now we see that those are the network neighborhood that are strongly enriched in stronger effect in proband compared to sibling. And those genes actually clusters into two groups. So one is synapse-related genes, another is chromatin regulation-related genes. And actually, both groups happen to have been the groups that has been previously indicated by the proband, by the coding mutations discovered in autism individuals. So this suggests a convergence of the mechanisms that are indicated by the coding and non-coding mutations. So I'll stop here and just go to my last slide. So thank all my collaborators that's being involved in these studies. And if you have any questions, also feel free to email me, which is here.
Great, thank you so much. And I saw that you were trying to go to your code earlier. Did you want to show off the?
Oh yeah, so we have a deep learning sequence model library that is actually designed to help you develop, train, and evaluate sequence models. And that's also the model the library that we depend upon, and we welcome you to use it and give us feedbacks.
Very cool.
The library is developed by a brilliant student, Cassie Chen.
And it has a very beautiful Greek name.
Yes.
The goddess of the moon. All right. Thank you so much, Jan. So I want everybody to tell me, do you guys feel that you learned something? Let's see. Awesome, awesome. I see a lot of applause. Awesome, look at that, 100%. This is really the best, 100%, 0000. This is great. Okay, awesome. Thank you so much, Jan, for a great lecture. And thank you all for sticking around. TAs, can I ask you to stick around just for a couple of more minutes?